Classical antibody discovery uses phage or yeast display to create large libraries of antibodies from which lead therapeutic candidates are identified. However, candidates with adequate binding properties derived from these platforms can have biophysical issues only identified in late discovery when they are manufactured in mammalian cells. For example, a tendency for the antibodies to aggregate at high concentrations can result in low production levels and increased development times and manufacturing costs. These issues can also generate unwanted side effects in patients, potentially leading to clinical or commercial failure. Scientists at Iontas have invented a novel technology for displaying antibodies on the surface of mammalian cells for antibody screening, which can detect and avoid antibodies with problematic developability profiles during the early discovery stage, whilst simultaneously deep mining antibody populations based on affinity, specificity, and species cross-reactivity. So how does this work? Huge libraries are created in the intended final format, typically IgG, directly in mammalian cell types used in manufacturing. Each cell has a single antibody gene inserted at a defined position in the cellular genome by an integrative nuclease system. This allows for the antibodies to be expressed under the control of the same promoter. Well-behaved antibodies are presented at a high level. If an antibody has poor manufacturing properties, copies come onto the cell surface and potentially aggregate. These aggregates are recycled back into the cell and degraded, resulting in few copies being displayed on the cell surface, whereas developable antibodies show high display. Using the power of multiparametric flow cytometry, many millions of clones in IgG format can be interrogated simultaneously for improvements in developability and binding properties, such as specificity, affinity, and species cross-reactivity. Iontas's approach has also been used to fix problematic antibodies and identify variants with similar binding profiles and better manufacturing properties compared to the original. Mammalian display therefore allows deep mining of antibody libraries in IgG format, reducing developability risk and shortening antibody discovery and development timelines. Learn more at iontas.co.uk.